Hello and welcome to today's webinar featuring Alysson Muotri. I'm Claire Cameron, Engagement Editor at Spectrum, the home for autism research news and analysis. We'll begin the presentation in a moment, but first, know that we'll be fielding questions at the end of today's presentation, but you can ask them at any point during the session. To ask a question, submit them via the chat window to the left of your screen. And as always, I'll note that if there are any members of the press tuning in, you can only report information presented during this webinar if that material has already been published elsewhere or if you have first obtained express written permission from the presenter. Professor Muotri joins us today from San Diego, California. Muotri joined the University of California, San Diego as a professor in 2008. He's an associate professor of pediatrics and of cellular and molecular medicine and a member of the UCSD Stem Cell Program and UCSD Moores Cancer Center. Prior to joining UCSD, Professor Muotri earned his BSc in Biological Sciences from the State University of Campinas and his PhD in Genetics from the University of Sao Paulo, Brazil, even. In 2002, he joined the Salk Institute as a Pew Latin America Fellow. He has received numerous awards, including the NIH Director's New Innovator Award, NARSAD, and the Emerald Foundation Young Investigator Award. Professor Muotri's research focuses on human brain development and evolution, exploring mobile elements as generators of neuronal diversity. Professor Muotri's lab is also interested in the modeling of neurological conditions such as autism using human-induced pluripotent stem cells. His lab has developed several techniques to culture human neurons and glia for basic research and drug screening platforms. Welcome, Professor Muotri.
All right. First of all, thank you all. Thank you, the Spectrum team, for organizing this webinar. I always found it very interesting, have seen some of them myself. So I'll be presenting some of the research that is going on in my lab, and I'll do it in two ways. So it's going to be a little bit broad, trying to cover lots of things, and packed with information. And I'm doing that because this is going to be recorded, so I think people will have the opportunity to go back and check if there is any information that was too fast, perhaps. Okay, so my lab really focuses on new models to study or to understand autism spectrum disorders. And we like to say that we come with a human perspective. And this has several reasons. Perhaps the most important one was really my frustration as a PhD student and a postdoc with mouse models for diseases that affect the human social brain, because these are really different from really what happens to humans, and the poor translatability of the animal model. So I thought that perhaps a human model, starting with human pluripotent stem cells, will give me the opportunity to learn something new, something that perhaps would complement the other models. So it was about the time that I was setting up my lab that the whole reprogramming technology come to life with Shinya Yamanaka. And what my lab does is really try to reprogram cells from specific individuals with genetic diseases and differentiating these cells into the relevant cell types, such as neuron or glia cells, and starting what we call a phenotype battery, looking for differences between a control group and an affected group. And these differences could be at the cellular level, such as neuronal morphology, could be at the connectivity level, such as how the neurons connect to each other, the number of synapses, the strength of the synapses, and also the circuitry level, how the information circulates into the circuitry. And if you're lucky enough to find one of those phenotypes, perhaps you could use that as a drug screening platform, looking for drugs that could rescue, revert, or ameliorate some of these phenotypes and moving fast to a clinical trial. So overall, I mean, this is the whole idea behind what we call disease in a dish. And what we hope to do is really to speed it up, the drug discovery part, so we can move into these clinical trials in a fast pace. When we start doing that, I mean, we were one of the first labs to apply that for disease modeling, and we decided that the safest way to do that was really to use conditions that are well genetically defined, and that was one reason why we start with Rett syndrome. So Rett syndrome is caused by mutations in the MECP2 gene. It's an X-linked gene that affects neurodevelopment. So it's a quite dramatic disorder. And here in this slide, I'm just showing some of the mutations that we have in both girls and boys affected with Rett syndrome. There are different types of mutations, and what we do is we reprogram these cells into pluripotent stem cells. We make cortical neurons, and we try to understand what is the difference, what's wrong with them. So because this was really early days at the time, we didn't know exactly what to look for. So we rely on the previous models. And previously, there was one description coming from a postmortem brain from a boy with a mutation in the MECP2 gene, showing that if you just look at the neuronal morphology, you see that the neurons derived from this brain are less arborized. The soma is smaller compared to controls. And this is true for different cortical layers as well. That's what we see in the top are the mutant cells. In the bottom are the representative images of control cells in the same brain region. So we thought that perhaps, neuronal morphology would be a way to start. And protocols to differentiate these cells into cortical neurons were just starting. We have been improving these protocols. This is just an outline of what we do in the lab. So we grow these cells in well-defined conditioned media as a pluripotent state. And then we use small molecules really to drive the cells to neural rosettes. And from that part, we mechanically select the best rosettes, and we dissociate them, and we grow them as NPCs or neuroprogenitor cells. We can follow up all of these steps using morphology as a readout. I mean, you can see how the cells will change in morphology, but also we have several markers such as SOX2 netting that will tell us that we are in the right stage. And from NPCs, you can stimulate the cells to mature into more functional neurons, and usually what we have is a mixed cultures. We have different subtype of neurons represented there, but also some glia cells and some other non-differentiated cells. So it's a mixed population. You can enrich that population for a specific subtype of neurons by magnetic or cell sorting, and this is useful for some of the experiments that we do at the biochemical level. So we characterize this population using immunostainings, as I previously showed, demonstrating that there are different cortical layers that are represented in there. But more recently, we have been taking advantage of the single cell analysis, where we can trap a single cell in one of these microfluid chamber and really do a more comprehensive transcriptional profile of these cells, and making sure that we have different layers that are represented, different subtype of neurons or neurotransmitters that are being expressed in the cell at a specific time point. So overall, I mean, we do that as a quality control, making sure that we are comparing apples to apples, and the differentiation has worked. So this slide, I'm just highlighting one of the very first observations that we made with Rett syndrome neurons. In the bottom, you have two mutant neurons here, and on top it's a control white type cell. As you can see in the bright field, these are mixed populations, so there are neurons in there mixed up with other cell types. And we use a genetic trick, which is a synapsin vector, a synapsin promoter driving a EGFP in a lentiviral vector. So we use this virus to infect the culture, so only the neurons would highlight in green, and you can observe the morphology. And when I was looking under the microscope, looking for phenotypes, the first observation was the soma size. If you get a random soma size here and here, you can see that in the mutants, the size of the soma measured by the diameter of the cell is really small compared to controls. So it's still a mixed population, so there are different cell types in there, but if you quantify hundreds and hundreds of cells, it becomes a significant difference. And you can validate that this is actually a true phenotype by using shRNAs that can specifically knock down the gene proving causation, proving that it's really the expression of MCP2 that's responsible for this morphological phenotype. So again, this was one of the first ones. Another one that was even more dramatic is the number of spines in this process in a dish. The human neurons will never mature as much as we see in primary mouse neurons or neurons isolated from the brain. So it's quite immature. So the number of spines that you get is not a lot. But even though, if you just quantify what you have, you see that Rett syndrome neurons, they don't have as many spines as in the control. And again, you can validate that by shRNAs. And at the functional level, we can measure the number of synapses by using immunostaining such as vGlut here and just counting the puntas that are close to this neuronal process. And again, as we would expected, Rett syndrome neurons have a decrease in the number of synapses. So these are glutamatergic excitatory synapses. But perhaps the most impressive data that I think this work showed was the ability to revert or completely rescue this phenotype by using specific experimental drugs. One of the drugs that we tried, IGF-1, if you just treat these Rett neurons with IGF-1, you can completely rescue the number of synapses after 2 weeks. So this is quite impressive and really open up the idea of a drug screening platform. If you do have a screenable readout, this is something that's very attractive to many scientists. All these defects in the number of synapses or connections we validate using electrophysiology. This is patch clamp electrophysiology. Again, validating or confirmation what we see in the immunostainings. So more recent, we have been using this technology really to recreate more complex three-dimensional structures in the lab, and this is what we call organoids or mini brains or 3D structures. And we have been playing around with different protocols that have been published in the past years, and we have our own protocol now that's a mix of these different good ideas that have been published before. And really we start with manipulating or dissociating single pluripotent stem cells into single cell stage. And from that stage, you can actually induce them to self-aggregate in these three-dimensional structures. We can call it this stage perhaps a neurosphere stage. And this will take like 1 or 2 weeks. And already at that stage, you can actually see that in those spheres you have different structures that you can quantify, or you can analyze those units. And those units usually have some more proliferative cells. So this is Ki67 here in red. You can see that many of those have rosettes formation with proliferating cells, but already some new N positive mature neurons that you can see at these early stages. If you just let these 3D structures really to self-organize and to proliferate more, you can keep them for a couple of weeks. And in our protocol, we see that these are very consistent. I mean, the size of these spheres is very robust. So this is something that we like a lot. We don't see a lack of reproducibility between batches of differentiation. So I think this is a really good advantage. And at this maturation process, we still continue to have some proliferative zones, but now we see that the neurons would migrate out from these proliferative zones and start to differentiate at the periphery of these units. Finally, you can keep those neurons to mature at even at longer time points, several months. We have them up to a year in the lab, and they would really continue to grow. After that, we see that some of the center of these spheres will actually die and become necrotic. But up to a year, they look fine. And they definitely grow in size. You can see that the size of the spheres will grow. And we try to follow up the human neurodevelopment, this is just a schematic to show different markers at different stages, and we try to match to see if this is really what's happening in our system. And just some of the images to show that it seems that they do recapitulate the human neurodevelopment quite well. We see both radial and tangential pattern of those neurons. And if you just do a time lapse on that, you're going to see that proliferative or neuroprogenitor cells would migrate out the upper layers. So this is quite beautiful to see. And as they mature, you see different subtype of layers or cortical neurons being formed, and those layers would become better or well-defined with time. Also with time, we start to see that those structures will start to fold, and we'll have these nice foldings all over these spheres. So we can, in a very subjective way, just trace some lines here and define different zones. What's the proliferative zone that contains cells that, neurostem cells or cells that are being dividing, or cortical plates with different subtype of neurons. So we do that in a tentative to quantify the different parts of this process. One thing that is very clear is the fact that compared to our 2D system, we do have more mature cells arriving at the same time. So this is CTP2 is one of the subtype of cortical neurons. In 8-week basic protocol with 2D, you have a couple of them here. But if you use our 3D protocol, it's really enriched by these different cortical markers. So it's a significant improvement. The fact that you don't see lots of MAP2 positive cells, it's because we have dissociated these cortical organoids. If you wait another week, the number of process here will be even higher than compared to the 2D protocol. So what I'm saying is that better protocols are coming every day, can tweak the protocol to have better neurons, better outcomes. And with this protocol, we can also use multi-electrode arrays to record from the networks that are being formed over time. So we just plate those structures on top of these multi-electrode arrays. And this is the kind of data that we get. It's a raster plot. So there's different channels being represented here. So each line is a different channel from the multi-electrode array. And each trace here represents one spike. So this is field electropotential. We are capturing the electrical activity that's passing through these networks. And as you can see, in 8 weeks, we do see some channels that fire more than others. It seems like a little bit random. After 10 weeks, we definitely see some kind of synchronization happening. And really at 12 weeks is when the networks are really mature. And you can see bursts of neurons that are synchronized. So this is really beautiful to see because you can keep those cultures alive and just keep recording from them. When we compare a group of control to our Rett syndrome subject, we see that although the electrical activity seems to be similar, the number of bursts that synchronize over more than 4 channels is really reduced compared to controls. So this was one of the first network observations that we see for Rett syndrome. What is really curious is that if you now work with another syndrome, and this is instead of having the gene mutated, you have a duplication in the gene, and this is called MECP2 duplication syndrome. We actually see exactly the opposite compared to controls at the same time point. Neurons that have twice as much MECP2, they are over-synchronized over time. So we still don't know exactly what does it mean. Does it mean that they are more prone to seizures, or how these networks will really correlate with human behavior? So it's still unclear to us. We are working on a way to translate what we find from these multi-electrode arrays to EEG recordings from the patients. But these are early days. We don't know yet how to do that. And as I mentioned to you, I mean, we can take advantage of these phenotypes really to find drugs. And for MECP2 duplication, this is another dramatic syndrome, and there is really no drug available. So we look for, we screen for about 45 drugs. These are epigenetic drugs, and looking for specific molecules that could revert these over-synchronized births. And we got some drugs. And what's interesting is that some of them could actually revert the morphology phenotypes, different from MECP2 mutants. The MECP2 duplication phenotype is the opposite. Those neurons are highly arborized. And these are just two of the drugs that we got, scriptate and NCH51. Both of them can make those neurons retract and behave similar to the control neurons. But it was only NCH51 that could rescue at the electrophysiological level. I mean, we could definitely see a reduction in the synchronization or total spikes after treatment with this drug. So this is a potentially new drug. It's a H-tag inhibitor, and we are moving into further preclinical testing to see if we can really take advantage of this drug in a clinical trial, in a clinical setting. I think perhaps one of the most amazing things is really the ability to take advantage of cellular phenotypes to screen drugs. And this was not easy. We took, just for Rett syndrome, like 3 years to test 3 drugs because the process was very manual. We didn't know exactly how to do it. But now, I mean, I think my lab and others are way ahead in the game, so we can do that in a more high-throughput fashion. In fact, we do have a collaboration with NIH where we are screening 55,000 drugs for Rett syndrome. And this is just some of the drugs that we have been screening. So these are candidates that can actually increase the amount of synapsing after treatment. So it's promising, looks good. We are now in the phase of validating some of those, and we hope that some of them would be in the category of drugs that are already FDA approved, so would be a more repurposing strategy than really a new drug. So summary for that part, I mean, we can make neurons from Rett syndrome patients and control patients, look for cellular molecular phenotypes, test drugs to see if we can revert or rescue some of those phenotypes. But the beauty of the model is that you can increase in complexity, and I already told you one example, which is to move from a 2D to a 3D approach. We can gain so much information. So another thing that we could do is really to explore the different cell types that are in a dish and see if we can dissect specific mechanisms, cellular mechanisms. One cell type that we are really interested in is are the astrocytes, these green cells here that really make the connections between neurons and blood vessels. Actually, if you take a drug that goes to your brain, astrocytes would be probably the first cell to receive the drug. So we think that these astrocytes are really important in keeping neuronal homeostasis. But the protocols to make astrocytes was really bad and took a long time. So we had to improve on that as well. So to make like a long story short, we also optimized this protocol. So we have a really nice, very homogeneous population of GFAP positive, S100 beta positive cells that we can get in 30 days with no addition of growth factors. And I'm highlighting the addition of growth factors here because if you really want to compare disease and controls, you try to manipulate the system as minimal as possible. And sometimes, I mean, just the addition of BDNF or GDNF may actually rescue some of the phenotypes that you are really trying to distinguish between control cells. So just an overview, the protocol is quite easy. We also start from neurospheres, and we move on into what we call astrospheres by changing the media to a more astrocyte-type media. And by plating these cells, the astrocytes would migrate out from these spheres. And if you remove these spheres, you can do it by suction. The astrocytes will mature and you have your nice population that you can propagate and pass 1 or 2 times. When we do that for Rett syndrome and we compare, this is the first data that we had by gene expression. You can see if you compare our controls in the black bar to the Rett syndrome population here in the white bar, you see that genes that are supposed to be high expressed in astrocytes, such as GSAP, aquaporin, S100-beta, vimentin, these are genes that are somehow overexpressed in Rett compared to controls. So perhaps one indication that Rett syndrome astrocytes, from the beginning, they are hyperreactive. Other factors such as BMPs and GDNF, these are factors secreted by astrocytes that contribute to a more mature neuronal network. These are downregulated in Rett syndrome astrocytes. And that might explain why the neurons look so immature. The other pathway that we see is totally misregulated is the glutamate pathway, where we see genes that are supposed to be up or downregulated in Rett syndrome compared to controls. So if you have a gene expression data like that, you can postulate some experiments. For example, if you take the glutamate pathway and if you just measure the amount of glutamate that these astrocytes can support, you can see in the black bar that if you challenge these cells with high levels of glutamate, these astrocytes will buffer that over 96 hours. Rett syndrome astrocytes cannot do that, so they will just accumulate these excess of glutamate, and that would become really toxic, so these cells will eventually die. Another thing that astrocytes do really well is to communicate through calcium waves, and they do that perhaps to inform the network that there is something wrong. So the assay that we propose here is really to mechanically stimulate a monolayer of astrocytes here in the red circle, and you can see how the calcium waves will propagate. So this is what a normal astrocyte will do it. It's actually really similar to a primary astrocyte isolated from a human brain. But Rett syndrome astrocytes, they don't do it. They will never propagate a calcium wave as much as we see in controls. So these are clear phenotypes that we could explore. But really, one thing that we would like to learn more is what's the interplay between astrocytes and neurons. And one way of doing that is to co-culture these cells in a dish. So here, what we do is just to purify some cortical neurons, and in parallel, we have this monolayer of astrocytes. We plate neurons on top of the glia cells, and we wait for 2 weeks, and we analyze the neurons. So what I'm going to show is just the morphology measurement that we had by mixing up the different genotypes. So this is how the cultures look like. In red, you have your beautiful monolayer of astrocytes, and in green, you have your neuron after 2 weeks. So we are looking for morphometric analysis, such as soma size, number of processes, and spines. When we plate control neurons together with control astrocytes, this is how it looks like. It's a very arborized, beautiful, mature neuron. If you now use Rett syndrome astrocytes and you plate control neurons, those neurons will not mature, which is consistent with the gene expression profiling that we see. The same is true if you have Rett syndrome neurons growing on top of Rett syndrome astrocytes. Those neurons will become immature, will never have like more than a bipolar look. But the most striking aspect is if you now move these Rett syndrome neurons, remember they have the MCP2 mutation, and if you plate them on top of control astrocytes, you can basically rescue everything. So all the morphology aspects we could revert. So it's quite striking because this is we are showing that there is a non-cell autonomous effect. Even though that neuron has a Rett syndrome mutation, they behave like a control neuron by being in the right environment. So this is important to us. And all these validations were done independently by a different lab. Here is just some of the quantifications that we have. But what could be the cause? What could be the factors that might be affecting these human neurons from Rett syndrome astrocytes? Again, if you look back to the gene expression analysis, what we have is really some dysregulation cytokines, and we actually validate that in our isogenic cells. So this is CRISPR-modified isogenic cells. You see that the levels of IL-6 actually go high if you mutate MCP2. And even in postmortem brain tissues from Rett syndrome individuals, you can actually see high levels of IL-6. IL-6 is an interesting molecule because it has been shown to impair human neurons in several models for autism, for example. And if you just add excess of IL-6 in normal networks, you can see that the morphology will change, the number of synapses will change, and the whole network activity measured by the number of spikes will actually change as well. So I think that IL-6 is perhaps one of the key cytokines implicated in neurodevelopment and in autism spectrum disorders. So everything I told you so far has been on well-defined genetic diseases. What about idiopathic autism? These are the vast majority. So we have been collecting tooth from kids with autism using this tooth fairy kit collection, where we mail the families package with a trial vial with a small medium in there, and we ask for the new tooth. So they send it back to us together with their clinical information. We isolate the cells from the new tooth, and we use these cells to reprogram. So we have a bank of those cells in our freezers, and we combine the information of the genomic information to learn something new. So this is one of the really first patients that we received cells, and after mapping, we discovered that they have a translocation by blocking a gene called TRPC6. So this is a gene that was really never implicated in autism before, was never shown in one of those large sequencing efforts to find genes for autism. So it was a new gene, and we didn't know exactly if this was causative or not. So we show that in different ways. So this is all published, so I'll skip some of these data. But we show that if you have mutations in TRPC6, you also have a lower number of spines, the number of arborization, or how complex those neurons is also reduced. And finally, you do have a lower number of excitatory synapses as well. So this all goes along with the data that I show you for Rett syndrome. And in the beginning, I thought that, well, I mean, maybe there is something unique here. Maybe MECP2 and TRPC6 are actually acting in the same pathway. And one way to demonstrate that was really to take advantage of the isogenic cells and measure TRPC6 level in both controls and Rett syndrome cells. And we see that if you have a mutant MECP2, you don't have as many TRPC6. So that suggests that both of these genes are acting the same pathway, and perhaps both drugs, both patients or individuals with mutations in those genes might benefit with the same type of treatment. We did the experiments in cells by treating the cells with IGF-1, and as we see in Rett syndrome, we could also rescue synaptogenesis in this individual. But what about something specific to this individual? So we thought a lot about that, and this is the concept that I like to bring, is personalized medicine. When you have specific genetic information from one individual and you try to find a way to personalize their treatment. And by studying the aspects, the biology of TRPC6, I found this interesting paper showing that hyperforin, which is an active constitutive of St. John's wort, can actually bind specifically to TRPC6 and activate calcium influx. And we thought that, well, I mean, if this is really true, we should rescue some of the morphology that we see in our neurons by treating the cells with hyperforin. And we did the experiments in a dish. So this is number of puncta, number of synapses. In the mutant cells, you have about 50% reduction. If you treat with hyperforin, you can rescue that. So suggesting that perhaps for those individuals who have alterations in the TRPC6 pathway, hyperforin might be a potential treatment. So finally, I would like to move with another genetic model. This is just to contrast what we have done with autism. And the idea is to use Weeden syndrome, who has a genetic deletion in the chromosome 7, and where the social phenotype is quite striking, because in this situation you have a hypersocial phenotype. And we thought that perhaps we could use Weeden syndrome cells to contrast to autism and learn something about the human social brain. So we generate iPS cells from 5 Weeden syndrome patients, including 1 atypical patient who has a small deletion here, and 4 control patients. And some of the phenotypes that we see, it is that there is no alteration in NPC proliferation. So the cells, they propagate at the same pace, the same ratio as the control cells. But we do see a high level of apoptosis on these neuroprogenitor cells, suggesting that about half of them will just die. I mean, they just cannot survive. So if this is actually true, if we do see a reduction in the proliferation or survival of Weeden syndrome neuroprogenitor cells, we should see a reduction in the cortex. So we team up with Eric Hallgren at UCSD, and he did some brain scans, MRI, to quantify the overall surface of cortex. And we do see, as we predicted by the cells, a reduction compared to controls, and this is true for different brain regions. So overall, Weeden syndrome brains are microcephalic, especially regarding the cortical surface area. But the cells that survive, they can differentiate into neurons, and then when we look at those neurons, we also see something that was not expected. So these Weeden syndrome cells, they're highly arborized. They make way more synapses than what we see in autism. Not only at the morphological aspect, but at the functional level as well. So this is just one of the examples with calcium transient to show that typical Weeden syndrome patients, their neurons, derived neurons, can actually have a high level of frequency or percentage of neurons that are firing the network. So suggesting that these networks are more active compared to controls. The way to validate that, of course, we cannot validate circuitries in a human brain, but we could take advantage of postmortem tissue. So this is postmortem Weeden syndrome brains, and we look into the exactly cortical layer, the subtype of neurons that we analyze in a dish, and we ask ourselves, can we reproduce that phenotype in the human brain? And as the iPSCs, the cellular data would point it out, these neurons are highly arborized compared to controls. So this was another surprise, and as far as I'm concerned, one of the few examples where the cellular model can actually instruct us or teach us something about the human disease that was not obvious, was not clear. And I'm glad we were able to validate that using other models. All right. So can as a final part here, my point is, can we use this model not only to understand the genetic aspects of neurodevelopmental disorders, but can we take advantage of that model and use environmental factors to learn something about networks, something about autism? And the way I got involved into that is because really I'm from Brazil. I have seen those dramatic images through the news, and I was really intrigued by the fact that the Zika virus, this outbreak of the Zika virus last year, could cause such a dramatic effect in the brain, such as macrocephaly. So I think most of you have already known that the Zika virus has been spreading to Latin America from the first appearance in Uganda. Then you see some of the detections of the virus in different regions of the globe until in 2015 it reached out to Brazil, and for some reason that we still don't quite understand. The virus become really aggressive, and we see all these cases of macrocephalic babies. When I was involved into that, there was lots of questions about, is really the virus the causative agent that is causing these macrocephalic phenotypes? We didn't know, and we proposed to test that in different ways. So again, this is a published study, so I will not go into the details of how we show that in a mouse brain, but I'll show that we can use these brain organoids really to understand a little bit of how the Zika virus can actually interfere with the normal formation of the brain. So we have been creating or taking advantage of these organoids and exposing them to the different strains of the Zika to see what are the consequences to neurodevelopment. So one of the most striking data that we have is when we analyze the different regions of these mini brains here, these organoids, and we see that there is a dramatic reduction in some of the cortical layers, and this is caused by a high level of apoptosis that you see in some of the ventricular zones where we have these neurostem cells, these proliferative cells. So the Zika is really causing these cells to die, and as a consequence, you don't have the right formation of these cortical layers. So this is really striking. We can actually play around with the different doses of the virus, and we haven't reported that yet, but there are many insights that you can actually get from just looking at those images. So there are instances, if you have a very low MOI, a very low viral dose, you can actually detect these small alterations that might not be very significant in the beginning, but if you let those brain organoids to mature and grow, those differences would become more dramatic. So this is just an alert to the fact that there are probably several kids that have been infected or babies born that were previously infected with the Zika, and they look apparently normal, but they may actually grow and have some problems at later stages. So it remains to be seen, but I think the clinical data, so most of those babies are now 1 year, 1.5 years old, and there are already clinical data pointing out to the fact that there are some visual defects, hearing defects, motor defects that are affecting kids even without the macrocephalic phenotype. So we'll see how the story evolves, but we are hoping that we can use this system again to screen drugs that can block viral replication or ameliorate this phenotype, especially if you can get that at early stages. So these are my take-home messages. I hope I'll convince you that both MCP2 and TRPC6 levels can both modulate the formation of glutamatergic synapses in human cortical neurons. Those two are actually acting in the same pathway. The IPS cells, or the IPS-derived neurons from both Rett syndrome and MCP2 duplication, both can successfully be used in a drug screening platform. You can actually use glia cells to do that as well. And Rett syndrome astrocytes have this impaired metabolism that is this inflammatory cytokine signature, probably triggered by these high levels of IL-6. We're still exploring that. What's the impact of IL-6 in these networks? But I think the good news is the fact that Rett syndrome neurons can be fully rescued by co-culture with wild-type astrocytes. That opens different ideas, different possibilities, even for cell transplantation. Would it be possible to transplant healthy astrocytes in a Rett syndrome brain to rescue those networks? So this is ideas that we are exploring in the lab. And then we use Weeden syndrome cells to recapitulate some of the insights that we get from a dish to the patients, and we do that using both postmortem brain tissues as well as MRI from Weeden syndrome patients. And at the environmental factors such as the Brazilian Zika virus, we can understand how the virus behaves in this setting, and also use as a tool for drug screening. So I think the idea is not only to repurpose drugs, but perhaps looking for drugs that can be neuroprotective for both human disorders and genetic and environmental causes. So this is what we're trying to do that now for the Zika virus. Finally, I'll thank members of my lab. I'm just a piece here. There is lots of people who actually do the work, and I'll not name them now because these are just a contribution for basically all of them in the lab, and my funding agencies. So I see that I already have my time here, and I'll stop here.
Hey, thank you very much, Professor Muotri, for that very interesting presentation. We have a couple of questions. The first one is, how many Rett patient-derived iPS cells or neuronal lines have you looked at?
In the first publication, we looked at 4 patients. We have now expanded that collection to include not only females, but males. So we have about 8 to 9 patient cell lines. 2 or 3 are duplications, and we also have a collection of CRISPR isogenic cell lines where we can either create artificial mutations or rescue some of the patient mutations with the system. So it's a large collection. As far as we can tell, they all behave very similar. We still don't see differences between mutation type or between the isogenic controls and the patient-derived cells.
How homogeneous are the phenotypes you described for the Rett cells across patients?
Very homogeneous. This is one thing that we are quite proud of it. There are a couple of phenotypes that we never reported because it was not consistent across cell lines, but the ones that we like the most are neuronal morphology, number of synapses, and the synchronization of networks. So these are quite consistent, independent of the genetic background. As I said, I mean, we can validate that using isogenic background. So it's not the isogenic background that is responsible for the variations that we have. So the variation is probably experimental variations, but it's not as higher as the difference between the mutants and controls.
Another question is whether or not there are longitudinal studies that are being planned with children who show signs of the Zika virus but don't have microcephaly to see whether or not they have further neurodevelopmental conditions.
There are studies ongoing in Brazil, as far as I know, and there are some already some clinical reports. These are being published in more medical journals. Some case reports suggesting that this is already happening. So there are the prognostic is not looking good actually.
One more question is, how did the phenotypes of the Rett cell lines compare to those described in the Rett model mice?
It is hard to compare because we are deriving cortical neurons. There are some reports on hippocampal neurons, which is a different part of the brain. I do believe that there are some of the phenotypes that seems quite similar, such as morphology or neuronal or soma size. So these are consistent across the different models. We see that in the iPSCs, we see that in the mouse models, we see that in postmortem brains. There are some of other phenotypes that are different between the mouse model and the human model. For example, there is a recent Hudazorbi paper showing like higher synchronization of networks in a Rett syndrome knockout mouse model. Again, I mean, she was looking into the hippocampus, but nonetheless, I mean, she sees a higher level of synchronization. We see exactly the opposite. We see that those networks do not synchronize. So are these differences because of the human genetic background, the differences between species? So we don't know.
Okay, great. So I think that's all that we have time for today. just to make everybody aware, a complete replay of this webinar will be available on the Spectrum site in the next couple of days, by Friday at the latest. It will also be published on our social media pages, so if you follow us on Twitter @Spectrum or on Facebook, then you'll be able to see the webinar there. The webinar is part of an ongoing series, so please check on our site for upcoming talks that are to be announced soon, and for video archives of previous webinars. Thank you again, Professor Muotri, for today's presentation, and thank you all for tuning in.
Thank you so much.